I'll open us in a quick word of prayer. Heavenly Father, thank you for another chance to come together and to look at your fingerprints on creation and how we can go about understanding how you created the natural world. Open our eyes to see your beauty and see your goodness through what has been made. We invite your Holy Spirit in this place. We pray in Christ's name. Amen. So starting off with a moment of science, this past week Vice President Pence announced the results from the reconvened National Space Council, which had not met since the presidential administration of George H.W. Bush. By executive order, the National Space Council was reconvened, which brings together not just NASA, but also the space programs of various military branches, including the National Reconnaissance Office, the Air Force Space Command, and some others. And the idea is to bring cooperation and reduce redundancies between what we really have are multiple space agencies within the United States, but for different purposes. And so one thing the Vice President announced was a return to the moon, which we have not been to the moon with astronauts since 1972. Greg?
I was curious if that council included some of the commercial
space
programs as well.
Yes, there's representation from SpaceX and Blue Origin. Yep. The last time we were at the moon with people was in 1972, and this is video from them doing field geology by some boulders, which are probably ejected from craters. On the left is Commander Gene Cernan. He has red stripes on his spacesuit. And on the right is the only PhD geologist to have gone to the moon, Dr. Jack Schmidt. He's on the right without stripes on his spacesuit, and they're analyzing some boulders there. Now, I'm really excited to be able to count Jack on the right here as a colleague. This is him last year giving a talk at a conference just down in Columbia, and that's also him on the moon. So, same guy. And I see him periodically at conferences. Lord willing, I'll see him at 2 conferences this month. And it's just, I've met Nobel laureates. It's cooler to talk to him.
So are we going to send people back to the moon?
That's the idea. Yeah. Since 1972, astronauts have only ever gone 1% of the distance to the moon to low Earth orbit. So we really pulled back away from exploration, and we're trying to get back into that. That's me getting to meet him a little left. So speaking of the moon, one of the projects I'm working on now in my postdoc at APL is trying to understand the ages of some of these large basins on the moon. These roughly circles are enormous craters that have flooded with flood lava, like the type that's in Hawaii. And those were caused by asteroids hitting the moon very quickly. And if the moon was hit, that means also Earth was hit. In fact, hit more because we have more gravity and a bigger cross-sectional area, so we're just going to get whacked more. But to age date these things, we need to do what we talked about last week, which is radiometric age dating. And so I'm focusing in on some areas where we may have some impact melt. When an asteroid hits a planetary surface, all that energy has to go into something, and it goes into 2 things: digging out a hole, but also getting super hot and melting a lot of the rock into puddles of lava. So we want to go back and study the cooled lava, and I'm just zooming in here on some areas where we could potentially send future missions, either with humans or robots. One example of that, again, that's Jack, lower right there. This is a boulder that rolled down from a hill on the moon, and they collected some samples from this boulder using a rock hammer. They broke out some pieces. This rock in the lab back in Houston is a rock right there. And if you cut it into thin sections and put it under a microscope and shine light through it, you can look at the minerals in there. And then you can go into those minerals and take out some of the isotopes and do radiometric age dating, what we talked about last week. And what that will do is tell us when these large basins formed on the moon, how long ago. Because our models for solar system evolution, if we plot the age of the solar system down here, where we're at the present at 0 years ago, and here's the beginning of the solar system 4.5 billion years ago, the impactor flux, that is how common asteroids and meteorites hit planets, started off very, very high, and it's been trailing off ever since, which is good because these things tend to kill things. So it's good that the impact flux has been decreasing. However, there's evidence from rocks brought back from the moon that there may have been, even though there's an overall decline, there was a spike called the lunar cataclysm, or alternately called the late heavy bombardment, that occurred between 4 to 3.8 billion years ago. This was somewhere between 20 to 200 million years in length, and represents a departure from the overall decrease. And what's interesting is that after this spike is where we have the first evidence for the appearance of life on Earth, the first isotopic evidence, namely that life likes certain isotopes of elements and not other certain isotopes, and that there's a concentration of these life-preferable isotopes starting at 3.8 billion years ago, as soon as this light-heavy bombardment or lunar cataclysm ended. And then the oldest fossil evidence for life on Earth is from about 3.5 billion years ago. So it's not clear if this actually happened, or if it was a series of spikes up here, or if it was just, or maybe it was just high for a long time and then it fell off right there. So we're not sure. We need more samples from the moon to be able to address that. This gets back to why the Vice President's announcement was important, is to give direction to NASA, is to go back to the moon, get some samples that we can actually use to test this hypothesis. In the background, it's just an artist rendition of what it might have looked like on the early Earth with the early moon during the light-heavy bombardment, lava lakes everywhere, impactors. It would have been really cool to see remotely. Yeah.
Explain to me real quick what the spike is.
So the spike was an overall decrease in the amount of asteroids and meteorites hitting planets, except for a brief increase in the impact flux. So more asteroids, more meteoroids, more comets hitting the inner solar system planets during a relatively brief window from 3.8 to 4 billion years ago. And the reason this is important is because life emerged as soon as this ended. So as soon as it was possible for life to exist, life did start to exist. And so understanding the impact history of the moon will get at the origins of life on Earth. So that's just an interesting question I like to think about. And so hopefully, at some point in the future, we'll have bases on the moon. So that's the moment of science. I want to go over a brief review from the previous 4 weeks. A lot of people have had to miss different weeks, so I want to go over it. The first thing that we really emphasized was looking at scriptural passages that show that God often works through intermediaries. God gets credit for something even though a person, an angel, or some naturally occurring phenomenon created that. One of those examples is in Genesis 1, where presumably Moses is writing, and the ground, the earth produced these living things. And then in the next verse it says, and so God made these things. The author isn't differentiating between something arising, quote unquote, naturally versus God doing something. God gets credit for the creative acts, even if there's a scientific explanation for that. Another explanation, or another example is how, is from embryology, how the Psalms say that God knit us together in our mother's wombs. That's very poetic language, and God gets the credit for our development, in utero development. But we also know through the science of embryology how that comes about. There's not a contradiction there. It's God by means of embryology in this example. So I would say that nature is an obedient and creative servant of God that carries out God's creative will in the universe. That we're not separating God's creative will from how we understand how things work. If this is true, then scientific explanations are absolutely impossible. They absolutely cannot put God out of a job. So we don't need to be, we don't need to be afraid of where the science leads. Last week we talked about radiometric age dating as a reliable measurement of an ancient Earth and solar system. We know how old Earth is from doing age dating of meteorites and moon rocks. The oldest meteorites aren't any older than 4.6 billion years old. And so Earth is slightly younger than that at 4.55 billion years old. We talked about how the decay rate of radioactive isotopes has been constant over the billions of years. One of the methods of confidence for that is from looking at faraway supernova through telescopes, knowing that light left those places 10 billion years ago, roughly, for example, and seeing evidence for a constant radioactive decay rate. So this cuts against the young Earth creationists' assertion that radioactive decay rates are higher in the past. Another reason is that if they really were higher, really so much energy would have melted the entire Earth, and then all age dates would have shown a 6,000-year-old Earth. So it's kind of a self-defeating criticism that young Earth creationists, I think, bring against this. We also talked about how we know how old the universe is based on the constant speed of light, distances, and how we measure distances to faraway galaxies and quasars in the universe through an overlapping series of, this is the cosmic distance ladder of an overlapping series of measurements, starting with radar in the solar system, going to stellar trigonometric parallax, which measures distances just the way our eyes can measure distances. And looking at secret variable stars that we can determine their distance based on how bright they look and how fast they're pulsating. And then ending up with type 1a supernovae, which can take us to the furthest places in the universe. And this all points to an expanding universe and the legitimacy of Big Bang cosmology. Again, this doesn't answer where the universe came from in the first place. It does answer, though, how the universe has evolved over the last 13.8 billion years. Most of the lectures are archived at my YouTube channel, youtube.com/NASAman58, if you like long-form YouTube videos, which I don't, but other people do. So that brings us to this week, talking about evolution and how this fits into an Orthodox Christian framework. For one thing, we've been lied to about what evolution is. I'm going to be using PowerPoint for most of this, sorry. I think the way evolution is typically taught is that it is natural descent through variation with natural selection. And okay, that's true, but it glosses over so many details as to essentially be wrong. And I hope to get at a lot of the truth of how evolutionary theory is. Even if it's wrong, this is at least how it's understood and practiced by practicing biologists. And I have to throw out the caveat there that I'm not a biologist. This is not as easy for me to talk about as some other areas in geology and astronomy, just because I don't have the formal academic training in this. I've been doing a lot of self-teaching, though, and reading a lot and looking at some peer-reviewed papers and trying to educate myself on this. But a lot of the same scientific principles are still the same as the types of science that I do. And so just in terms of being scientifically consistent, I have a bit more confidence in tackling biology and biochemistry. So the first part, natural selection, is just the death and extinction of organisms that don't work. They have some body plan or some body chemistry that does not favor them to reproduce. And so if they die before they get a chance to reproduce, their genetic material is removed from the pool of available genetic material, and they don't pass on their genes. Basically, it's the idea that what is left over is good enough. It's the same idea, I suppose, in architecture. The old buildings that still exist, they still exist because they're structurally sound and they're good looking. If they were ugly or structurally unsound, they fell down or were torn down. That's a form of natural selection of architecture, for example. Another idea that's often tossed around is that mutation is random. That's just a straight up lie, as we'll talk about throughout the rest of this class. A lot of criticisms against evolution is that mutation that happens in the genetic material of cells is always deleterious and harms the organism. And they're talking about random mutation like a copying error. As DNA is being copied, as a cell is reproducing, some mistakes can be made, and some of these mutations might be beneficial for survival and can lead to different adaptations of the organism. And also the idea that some mutation is caused by cosmic ray hits, where you've got energetic charged particles that knock out parts of our DNA and lead to mutations. And all these types of mutations do happen. The fallacy is that this can ever be beneficial. Information theory for mathematicians shows that this is essentially always harmful to the organism. So mutation that is beneficial has to happen some other way. And the way practicing biologists is what we'll talk about, and how practicing biologists deal with this is shown throughout. I want to make sure this doesn't stop recording. Okay. So just as a review for all of us, all several trillion cells in our body contain cells with a nucleus in the middle with DNA inside the nucleus. The DNA is itself a very long molecule that has this double helix structure. And actually, it's a copy of the DNA. And each single strand of this is identical to the other strand that it's combined to. And they're connected, but they're connected head to tail, such that the start of one is at the ending of the other, but they otherwise contain the same information. And then the chemical bases made up of cytosine, adenine, guanine, and thymine, make up the information that code for an organism, in our case human. It codes for certain traits, 5 fingers, fingernails, wavy brown hair, blue eyes, male, etc. And all that information is encoded by combinations of these 4 chemicals. That's pretty amazing to me, that you can encode that much information with those 4 chemicals. And so biochemists abbreviate this with the first letter of each chemical, C, T, A, G, and T. C, T, A, and G. And then during cell reproduction, the DNA spools into like an X-shaped chromosome. That's not the same as an X versus Y, but they just kind of make an X shape. So we'll go into some of the evidence for historical evolution. And this is primarily based, well, it started off in geology, realizing that different organisms lived at different points in the past. This is incontrovertible. You really can't get away with saying, I don't, refuting this. We know this. Organisms go extinct. The fossilization of an organism is the exception, and it's by far not the rule. For fossilization to occur, you need an animal to die, or a plant to leave its imprint on a rock. You need some preservation to happen, usually one that's poor in oxygen to prevent decay. You have to make sure that scavengers don't go after it, eat it, scatter the bones everywhere. You want there to not be a landslide or earthquake that shakes things up and moves things around. You want there to not be a river that goes and washes things to the ocean. There have to be really good preservation environmental conditions for fossilization to happen. So where we do see fossils, that's where the environmental conditions for the past, however old the fossil is, up to literally billions of years, have been favorable for the preservation of that, and that is by far very rare on Earth's surface. As such, there is an incomplete fossil record, and we should expect to not, and there's probably many organisms that lived, died, and went extinct for which there's no record. One of the ways we know about this is from looking at stratigraphy, layers of rock in the rock record. This is a view of some random canyon I flew over a week or two ago out west. If we zoom in, we can start to get an idea of the layers of rock in there. I think it's just a beautiful view. There's a bridge on the lower left. Of course, the poster child for this is the Grand Canyon, where you can see different layers of rock. There's a red sandstone unit here. There's different limestone units going up through here. And different fossils are found in different layers. The oldest stuff is at the bottom. And as you work your way on up, you get younger and younger rock. And that's borne out through radiometric age dating of those different rock layers. And you see different types of fossils. And those fossils don't overlap. For instance, here's a view on the left. I took up the Grand Canyon. It's gorgeous. This is at sunset, so the reds are exaggerated. But so the Colorado River's on the bottom, and as you move up, you've got sandstone, shale, limestone. Not sure what this is. Shale, sandstone, and a different type of sandstone. And different fossils are found in those layers. So you might see trilobites on the bottom, and then trilobites, which were kind of like horseshoe crabs, but they lived 500 million years ago. They went extinct about 300 million years ago. And different fossils are found in different layers, and there's no overlap.
Can you explain that? How could there not be overlap?
Well, so typically extinction events are periods in Earth's history where there's violent and quick climatic change. It can be a supervolcano. In Siberia, there's what are called the Siberian traps, huge extensive flood lava that would have released enormous amounts of carbon dioxide, lots of ash, covered Siberian lava. This goes to very quick climate change. And a lot goes extinct very quickly. And so when things go extinct, they can't, that's it. So the reason there isn't overlap is because organisms go extinct and new species at least appear, I would say evolve, but at the very least they are created or appear and can continue on until they go extinct. So something that's very old, like crocodiles. Crocodiles have been around have overlapped with dinosaurs. They haven't changed much in 100 million years. And why should they? They're really good at reproducing and killing anything that wants to eat them. So there's no evolutionary pressure on them for them to change. So you might see crocodile fossils that would overlap down here, but they're not extinct. If they were extinct, they would stop at some point and then not overlap with stuff that's younger. So that's a good question. This is in contrast to what's portrayed at the Noah's Ark exhibit in northern Kentucky, where they have dinosaurs in the Ark. Humans and dinosaurs did not live at the same time. There are no overlapping fossils between dinosaur bones and human bones. All dinosaur bones are found to exist at layers that correspond to 65 million years ago and older, back to, I think, around 250 million years. Yes?
So is that exhibit a young Earth?
Yeah.
That's what we're talking about.
Yeah.
Their theory is young Earth. They're behind that.
Yeah. So this is put out by the Young Earth Creationist group Antigenesis, led by Ken Ham, where a lot of what I've been talking about would refute a lot of the things they claim. And so they believe that dinosaurs and humans overlapped. There's literally no evidence for that.
Even woolly mammoths.
Oh, woolly mammoths lived much more recently than dinosaurs. Woolly mammoths and dinosaurs didn't overlap.
So they're not dinosaurs.
No, they're not dinosaurs. In fact, humans and mammoths did overlap. In fact, the reason they went extinct is because our ancestors hunted them to extinction. The last mammoth died, we think, around 8,000 years ago in an island north of Siberia, where there was, in fact, it might have been more recent. It might have been only 6,000 years ago. But there was an island in northern Siberia where there was, we think, the last population of mammoths took their last stand. Now, they were all interbred with each other because there was no other mammoths to breed with. And so there's a lot of mutations that had developed over time. And so genetically, they were very weak, didn't have the genetic diversity. And so they would have gone extinct fairly easily. But, so woolly mammoths lived from a few 100,000 years ago up to a few thousand years ago. Yeah, but they didn't overlap with dinosaurs either. And our bones do overlap with their bones.
Right.
Now, evolution predicts that there ought to be transitional species. One thing that the Antigenesis folks assert is that we have not discovered any transitional fossils, And I don't even know how they get away making that because we have tons of them. We have examples. One example is Archaeopteryx. It's thought to be one of the first quasi-birds. And it was related to theropod dinosaurs. Theropod dinosaurs were dinosaurs that stood on 2 hind feet. So like T-Rex, Deinonychus, Velociraptor are common examples of a theropod dinosaur. And evolutionary theory, and we know from evolution that birds are the direct evolutionary descendants of theropod dinosaurs. So dinosaurs didn't completely go extinct, they're flying around. What is consistent with the idea of transitional species are fossils like Archaeopteryx. You can see imprints of the feather. So this rock is probably shale. You can obviously see the bone structure. You can see where the arms have evolved into wings. And you can see imprints of the feather throughout here. So this is an artist's reconstruction of what it could have looked like. Definitely a cross between a dinosaur and a bird in this case. And so this is an example. So this is at least consistent with the hypothesis that you would expect transitional species. Now, if I could briefly argue a young Earth creation perspective, you might say that, well, God just made this animal and not anything, and it wasn't related to anything before it or after it. Yeah, sure, maybe, but this is at least consistent with the hypothesis of transitional species. And we have more examples of transitional fossils in the form of whales. And you can see a lot of the stuff if you go down to DC, to the National Museum of Natural History. They've got examples of this. They've got a whale skeleton. And what's interesting about a whale skeleton is this is the pelvis. It's not attached to the rest of the skeleton at all. When they mount the skeleton, they have to have like strings suspending it below the spine. Our pelvis is definitely connected to the rest of our skeleton. But a whale's isn't. In a living animal, that pelvis is just surrounded by soft tissue. And we've seen fossils that are consistent. Oh, and the idea in evolution is that whales evolved from land animals about 50 million years ago. So this is 15 million years after dinosaurs. That you initially had, this is Diasodexis. This was a land animal with hooves. And over time, its body plan changed. It appears to have changed to one that was more adapted for swimming. It would spend less time on land and more time in the water, and its nostril would migrate from the front of its skull up to the top to become a blowhole. And these skeletons are consistent with that. I think, so in this one, the nostril's at the front here. Here, there's a bump here where the nostril has moved kind of closer up there. And I would imagine that these skeletons, the nostril would be even further up on the skull, allowing for a blowhole. And you can see how the hind limbs and the pelvis are becoming smaller and smaller, while the rest of the animal becomes more and more adapted for swimming. So yeah, it could be the case that maybe God just snapped his fingers and these animals appeared at different points in Earth's history. In fact, that's what the old Earth creationist group, Reasons to Believe, teach, that somehow animals appeared out of nowhere by God's direct involvement without using any intermediaries, without any predecessors, and they appeared. However, evolution predicts this, and this is what we see. So these are consistent with evolution, even though they don't prove it, they're still consistent. Yeah, Andrew.
I'm under the impression that sea creatures evolved first and then became land creatures.
That's true. This is one example of going the other way. So 50 million years ago is actually pretty recent in Earth's history. We had mammals. 50 million years ago, we had advanced mammals. We had advanced reptiles, fish, birds, everything we have today 50 million years ago. And yes, the first land animals were amphibians that came onto the land. But this is just one example of going the other way. Once we got on land, grass is always greener on the other side, so I want to go back to the water. Okay. So these are called vestigial appendages, or vestigial parts of the body. This pelvis is basically functionless. You could probably surgically remove this from a living animal and it'd be fine. There's just no reason, it just hasn't gotten completely rid of it yet. Yeah?
Is a coccyx a vestigial tail?
Probably. I think that's correct, yes. This is just another thing, shows the same thing. Again, a pelvis on a skeleton being suspended below the rest of the skeleton. But where I think the real evidence and the real power of evolution isn't just in the fossils, it's in our genetic code. Code is like, our genetic information is, it contains information, it is analogous to computer code. It is, it's amazing, and we're going to talk more about that. One of the evidences taken as, or some of the observations, these are the observations. The interpretation is then evolution, but the observation is that humans, chimps, other apes, and monkeys we're not able to produce our own vitamin C. We have to get vitamin C from our diet. A lot of other animals can produce their own vitamin C. Now what's interesting is that the place in our genetic information that is responsible for the deficiency in producing our own vitamin C is the exact deficiency in all of these other animals. It's in the exact same part of the DNA, and this keeps us and other apes from producing our own vitamin C. Now, apparently guinea pigs and fruit bats can't produce their own DNA either. However, the place in their genetic code responsible for that vitamin C deficiency is different from these. So that's an observation. Even a younger creationist would be forced to agree with that observation. The interpretation of that observation, then, is that we would share a common evolutionary ancestry with the other apes, other animals that have the same genetic mutation that we have, suggesting common descent. By the way, evolution does not say that we evolved from monkeys. It says that we and monkeys and other apes evolved from a common ancestor that lived a certain time period away. So, there's that stupid critique of evolution. I didn't evolve from a monkey. No, you didn't. You and monkeys evolved from the same common ancestor. So that's just kind of an aside. We also see vestigial genes in our chromosomes. So humans have, 26 chromosomes? Chromosome pairs. Apes have one more. And if you look at human chromosome 2, and every chromosome, I am taught, has what are called telomeres on the ends of the chromosome that are they're kind of like the stop sign. They're like, all right, this is the end of the chromosome. It's like a molecule that's hanging out on the edge there. So this is the end of the chromosome. And the middle of the chromosomes have what's called a telomere. And I think it's the central bit in the middle, I think. Someone with more education can correct me. What we see in human chromosome 2 is we see a telomere in the middle of the chromosome, which is normally, in every other occurrence, is on the end of a chromosome. And there's also 2 centromeres within the chromosomes. And this is exactly what you would expect if 2 other chromosomes had fused at some point in the evolutionary past. And indeed, that's what evolutionary theory holds, and that's what the evidence teaches. Telomeres in this example are compared to like the plastic bits on the end of your shoelaces to keep it from fraying. Same idea, I guess, for telomeres. Again, I'm a geologist, not a biologist. So, but this is very compelling evidence that our common ancestors with apes, there was a line there where the chromosomes fused to become our human chromosomes 2A and 2B, or what were there in apes. In our DNA, and this is a result from mapping the human genome starting in the 1990s, from mapping the human genome, we also see that we have the genetic code in our DNA for encoding for egg yolk protein, except that we don't lay eggs to reproduce. And it was found by knowing where it was from sequencing chicken DNA and saying, okay, if it's here in chicken DNA, then it should be here in human DNA if evolution is true. And indeed, that's where we found it to be. What this points toward is us and chickens sharing a common evolutionary ancestor 310 million years ago to an egg-laying reptile. These are just observations, and the interpretation is evolution through common descent and mutation. So I want to get into now how this mutation works. I've been saying it's not random, it's not random. Now we're going to get into talking about how it's not random. The acronym I've come up with for this is THESH, Transposition, Horizontal Gene Transfer, Epigenetics, Symbiogenesis, and Hybridization. And these, we're going to touch on each of these in turn. But if you learn these words, and you can, I just invented this, it's not a real word, but at least you can pronounce it. And the idea is that mutations produce the changes from which natural selection selects. And again, natural selection is letting things die that are not useful for the organism and allowing things to reproduce that do have favorable mutations in there. And I have to say that I got essentially all of this from the book Evolution 2.0 by Perry Marshall, who's he's an applied mathematician and a computer scientist who's been applying this to biology. And this book is really informationally dense. It references a lot of peer-reviewed papers, and some of which I have gone back and fact-checked. I've looked up the peer-reviewed papers in the journals, and I've never found him to be wrong in this. So, I have some confidence that he knows what he's talking about, having gone through some of the literature myself. The bottom line, or one of the bottom lines I think that I'm going to get back to, is that evolution is easy. This shouldn't be the controversial thing at all. What's hard is where the original code came from for DNA. That's the hard part. Not how species change and evolve over time, but where does the original code for DNA come from? Because we don't have to my understanding, we don't have a theory for how information arises in the universe. So let's deal with the first T, transposition. This work was done by Dr. Barbara McClintock, who got the 1983 Nobel Prize in Medicine. And she did a series of experiments on corn, on what I was grown up and taught to call Indian corn. And it's a hard stuff with lots of different colored kernels on there. And she would go in and she would make deletions and otherwise mess up the DNA in the corn plant. And she could track this by the pattern and colors of the different kernels. And what she realized, when she would damage DNA, almost all the time the cell would go back and fix the DNA. And if it couldn't fix the DNA the way it was before, it would at least fix it in a way that would allow the corn plant to continue to live. And so she could change the order of these things. And what she realized is that even when you have mutations, the cell will go back almost 100% of the time and fix those mutations. So this goes against the way we were typically taught evolution in science documentaries and in arguments, because this stuff isn't really taught that well in school. And so people only have this cartoonish idea of what evolution is, and then they go back and refute the cartoon. So it's like we're set up from the beginning to do a straw man argument.
So Kirby, these cells will fix on the next cycle of reproduction? Is that what you mean? Like the seed of those things will fix?
No, even within the cell, even within the seed, those cells would go and repair their own DNA that Barbara had damaged on purpose. But when she was successful in damaging it, she noticed how the corn could adapt to that and make a repair that maybe not put it back exactly the way it was, but to make it livable and able to still reproduce typically. And she recorded this by painstakingly, again, looking at the pattern distribution of different colored kernels. And she was laughed at and derided, and like her work was censored from journal articles for a long time, until finally the rest of the scientific community caught up with her, and she was vindicated by getting the Nobel Prize. Now, the way transposition works is if you have a set of code, we're going to represent this with letters and numbers, A, B, C, 1, 2, 3, D, E, F, G, H. Transposition is a cell's mechanism where proteins within the cell can transpose sections of code, literally like moving blocks of code around in a computer program, such that instead of having this, you could then rearrange it to A, B, C, D, E, F, 1, 2, 3, G, H. It's just taking this section of code and moving it later in the sequence. This is one way that we can mutate the genetic information. Another mechanism is horizontal gene transfer, and this is responsible for antibiotic resistance. And I like this cartoon. It says, it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. Hey kid, want to be a superbug? Take some of this into your genome. Even penicillin won't be able to harm you. And this is a real problem. As doctors overprescribe medications, and as or antibiotics, and as ignorant patients demand of having an antibiotic for their viral infection, which will do nothing, they are creating evolutionary pressure on bacteria to get antibiotic resistance, to the point where we'll have bacterial infections that we can't treat. People will die from that. So it's scary. Now, the way horizontal gene transfer works is two cells will saddle up next to each other. I should actually go to the next slide first. Two cells will saddle up next to each other. One might already have a mutation to allow it to be resistant to antibiotics. The cells then create a pilus between each other, and this cell will send over a copy of its DNA. This cell then, through its reproduction mechanisms of messenger RNA and ribosomes, will then replicate that information, and now both cells are antibiotic resistant. But it's not just antibiotic resistance, it's other forms of information. Two bacteria can and they can be of different species, they don't have to be the same species, can share genetic information with each other through a pilus and exchange beneficial genetic information. Greg?
Would that be the same mechanism that the cells are using to repair damages in their DNA?
It could be one way, but not necessarily the only way. I mean, the way cells, I think, typically repair damage is just with little proteins within each cell that are their job is to do that. Typically, every time, so with 3 billion base pairs in each of our cells, I think there's typically one mistake per 10 million, roughly, and so that means that there's about 1,000 errors every time your cells reproduce. However, essentially all of those get corrected after the DNA has been copied through just onboard mechanisms within the cell, is my understanding. But this is a way that different species of single-celled organisms can exchange genetic information. So this is a way. So normally genetic information comes from parentage and reproduction. This is a way of getting information from another species. So this is modification of DNA, not through direct descent. Andrew?
So, really quick back to the transposition. Why would it transpose?
If Barbara McClintock had damaged the DNA, and the cell's like, aha, you damaged me, but I'm going to fix it by moving this stuff around.
Oh, okay.
And
It's just a way to fix it.
it's just a way to fix it.
Okay.
So when there are transcription errors, or there is like a cosmic, like radioactivity knocks out some, like radiation damage will damage our DNA, these are ways that cells can try to survive, is transposition.
Okay. So by fixing itself, that's kind of proof that it's not random.
That's part of it, yeah.
Part of it.
Going back to the previous slide here, just a way to conceptualize this is that if two different organisms, say two different species of bacteria, one has A, B, C, D, E, the other one has these letters, horizontal gene transfer just says that we copied these, didn't remove them from here, we just copied them, and now this code is also in this code of this organism. Now this organism has a genetic code of both. And whatever advantages this had for surviving in an antibiotic-resistant environment or whatever, or an antibiotic environment, this thing now has the ability to do also without changing species.
Does horizontal gene transfer only happen like with bacteria or more complex organisms?
It can happen in complex organisms too. So my understanding is that this has potential to happen in our own gut. We have an ecosystem of bacteria living in our intestinal tract, and they can exchange information with our cells too. So my understanding is this is also possible with multicellular organisms. I think it's more common though with single-celled organisms. Yeah. So this is a schematic understanding of how it works. Apparently, it can take as little as 20 minutes for this to happen.
Kirby,
Yeah.
when we think about new medical technology and genetic engineering, for example, to remediate like cystic fibrosis as an example, I'm sure I'm mixing two different events here, but why wouldn't then, if the cell has the ability to sort of correct all that, why wouldn't a cell look at genetic engineering and say, you're hurting me, so I want to correct for that and rule that out and make that not possible?
That's a great question, and I guess I don't know enough to be able to answer that, except that if you do a good enough job with the genetic engineering, there probably isn't a perceived need to change it.
So it
would be
would be
my
perceived
guess.
as damage.
It wouldn't be perceived as damage, right? I guess.
Okay.
What this means for the tree of life in terms of how things descend is that, to use a dirty metaphor, life is promiscuous, and we're exchanging DNA in ways that are not related to reproduction. So we've got branches of the tree of life touching branches of other parts of the tree of life, moving around genetic information, allowing mutations to happen, many of which can be beneficial to the organism. Another way that mutation happens within evolution is in epigenetics. And this is not changing the code, but it's switching code on and off. Or if you do computer programming, it's like commenting out a block of code. Or uncommenting out a block of code to allow that code to execute. The example, this was seen in studies from the Dutch famine of 1944 to 45. This was an industrially developed country where people were literate and typically had enough food, except that during World War II, the Nazis cut off their food supply, and people were starved and malnourished. And it didn't change their DNA, but the people who were affected by that starvation, their genetic code turned on and off certain parts of their code to allow them to survive in a low nutrient environment. Their offspring also had genetic adaptations to allow them to survive in a low nutrient environment. People tended to be smaller. They also were prone to more diseases like diabetes and other things that they were adapted to survive in a low nutrition environment, but there were some deleterious consequences to that. And this is an example of epigenetic changes. This epi means beyond. This is beyond genetics. We're not talking about changing code. We're just talking about expressing or not expressing certain genes. This would be similar to, in this example, of graying out lines or deleting lines from a text here. So this example given by Perry Marshall in his book is Flight 6429 was delayed from Chicago to Winnipeg, so it will not be departing before 6:45 p.m. From an epigenetics perspective, you can not delete, but actually just not express certain code. You can gray them out, such that you can make it say, Flight 29 to Winnipeg will depart before 6. Again, we didn't remove or add information, we just expressed or not expressed certain amounts of information. And this, by the way, what we've been talking up to now, but we'll continue talking about the transposition, the horizontal genes transfer, and the epigenetics, they're not random because they follow rules of chemistry and biology. Just like these are following rules of English grammar, We are not randomly crossing off letters in this example. We're following the rules of English grammar. We have a sentence that is grammatically correct. It's not just random letters floating around as you would expect from the way evolution is taught of just crossing off random things. It follows certain rules, so it's not random in that sense. The transposition, the horizontal gene transfer, and epigenetics are following certain rules that you can't predict ahead of time, but they're not random as we would understand random. The S in fesh is symbiogenesis. It's two things becoming one thing. One example where there's not a complete version of this is in lichen. If you go hiking and you see like these white or orange or yellow splotches on rocks, this is an ecosystem of fungus and algae living together. My understanding is that the fungus can protect the algae and serve as a source of nutrients for the algae, and the algae helps anchor it to the rock so it's stable. A number of papers and evidence have shown that this is responsible for a lot of the organelles, a lot of the small structures within cells, especially, let's see, our ribosomes in animal cells and chloroplasts in plant cells. So in an early plant cell, cyanobacteria, which is the very first life on Earth, got inside a cell. Maybe the cell is trying to eat this, except that once inside, the chloroplast started doing photosynthesis and started feeding its host cell in a symbiotic relationship. It then got to the point where this cell became dependent on the chloroplast and the chloroplast dependent on the cell, and it became a permanent part of the cell. In fact, chloroplasts in all plant cells have their own DNA. Most other organelles, again, they're many versions of organs, but within each individual cell, don't have their own DNA. They are encoded for by the DNA in the nucleus of the cell, whereas chloroplasts and then, it's mitochondria in animal cells. Is that right? Mitochondria? Is that right? Okay, cool. They also have their own DNA for reproducing. So this observation is taken as evidence for evolution and common descent. A cool example here, this is from a paper by Margulis in 1971, and it's been talked about since, of what he called serial symbiogenesis, of development of modern complex cells from the incorporation, just like literally sticking other cells inside other cells at some point in the past. Where if we want to trace the development of a modern plant cell, you start with a eubacteria and an archaeobacteria, really early primitive life, merging in a symbiotic relationship to give you an archaeoprotist, where the archaeoprotist and a paracoccus merge into a symbiotic relationship, where at first they're just kind of benefiting each other, but then they get so interdependent that they literally become the same organism, turning into a protozoan, which you can see under a microscope. And then a protozoan absorbing cyanobacteria and becoming algae, and then algae in yeast cells forming symbiotic relationships and becoming a plant cell. Similarly, you can start off with a protozoan, you can incorporate the eubacteria up here, and end up with an animal cell, like a paramecium. So, again, this is called serial symbiogenesis, the evolution of the early cells of life. Where just schematically, let's see, whatever A is just gets incorporated into B, and these things merge to form more complex cells that benefit all parts of it. One sub-example of symbiogenesis that should be RNA, from retroviruses. Now retroviruses, probably most people are familiar that a virus will go and infect a host cell, it'll stick in its RNA, its ribonucleic acid, literally reprogramming that cell to only make copies of the virus. And so the cell will do that. It'll make copies of the virus, which then explode the cell, and the other virus particles with their genetic information go on to infect others. The way a retrovirus works is that it injects its RNA or its DNA into the host, in this case a mouse, where instead of reprogramming the cells to just make copies of the virus, it takes its chunk of DNA, it splices apart the animal's DNA, and it sticks its own DNA right inside there. The animal, it could be killed by this process, but the animal can otherwise just go on living its life, except that the reproductive mechanisms are now also replicating the information that came from the retrovirus. And there are times when this retrovirus infection can be beneficial to the animal, and it can enhance its chances for survival and give to new body plan changes and promote the evolution of this animal by changing its programming, by just inserting new code that wasn't there before. And this is all of these examples: transposition, horizontal gene transfer, epigenetics, and now symbiogenesis. These are all observed in the lab. None of this is controversial. Like, practicing biologists observe this. So when people say we've never seen evolution happen, sorry, there's thousands of papers that show this happening in the labs, in biology labs all over the place.
Is all this stuff happening simultaneously too? I mean, these are not, are these discrete? What do I want to say? These are discrete, these 5 letters are discrete, but they're all happening right at this moment.
Yeah, I mean, they can happen by themselves, I suppose, but there's nothing to, but you can have a retrovirus. You could,
No, I don't mean the same action within the same cells. I just mean these are, all 5 processes are happening independently, but simultaneously.
Yeah, that's correct. Yeah.
Is there an example of how this works in humans?
I'm sure there are.
Okay, I'm just trying to make a connection. All right.
I mean, humans, just speculating off the top of my head, I mean, humans could be infected by a retrovirus, and it would probably kill us in most cases, but once in a while there will be an infection of the retrovirus that might give us beneficial survival at different latitudes. So if we look at our own human evolution, people of Asian descent have light-colored skin, people of Northern European descent have light-colored skin, but they have light-colored skin for different reasons. And it's to live in northern climates where they have trouble producing their own vitamin D from the sun. And so they need less melanin in their skin to let in more sunlight. And so the changes that allow for that could have happened from a retrovirus. They could have happened from other mechanisms also. But that's an evolutionary change within humans. Now, that hasn't happened to the point where where people of different ethnicities can't reproduce with each other, but they are distinct genetic differences. So I think that's a case of convergent evolution, where people of Asian descent and Northern European descent, both having light-colored skin, came about it from different ways, in different processes, different mutations. And the manifestation of that is actually different in both ethnicities, which I think is really interesting. Oh, same for dark-colored skin for people of African descent and Southern Asian Indian descent. They have dark-colored skin, both to protect them from the sun, but for different genetic changes occurred to allow that to happen. I think it's just really interesting. Again, hybridization. Most of the time when we think of hybrids, like a mule, a horse, and a donkey, yeah, they can reproduce, but they can't produce sterile offspring. Therefore, the things that had sex with each other aren't the same species, because they produce infertile offspring. However, once in a while, fertile offspring can result from the copulation of 2 different species. It's very rare, but it can happen. One place this might happen is during times of violent climatic change. Say a stream is drying up, and a given fish can't find one of its own species to mate with, so it'll mate with the closest thing it can find. Usually that won't result in anything, but once in a while it will. And this is where we take genetic information from 2 species, we copy them, and we have A, B, C, D, E, F, and A, B, C, D, G, H from both organisms, combining into a supergenome for a new organism. And this is indeed what has given rise to the animal, the hagfish, where tunicates of 2 different species, both with 14 chromosomes, apparently mated, and produced a hagfish with 28 chromosomes. They got lucky and produced a viable offspring that could reproduce. My understanding of hagfish is that if you bother them, they'll secrete slime to get away.
Great.
Yeah.
And so hagfish can now reproduce with each other.
Yeah.
Okay.
And they don't apparently mate with tunicates anymore.
Interesting.
Yeah. Another example, which I think is incredible, is 2 jawless vertebrates, both with 28 chromosomes, creating a jawed fish. Neither they might have spines, so they'd be chordates what am I thinking of? Not chordates, but anyways, they have spines, vertebrates, but not jaws. But the information was there. It was latent, but not expressed for a fish to have jaws. And these are examples that we've seen of two species through hybridization creating a new species. I think that's incredible. This is another mechanism of mutation that allows for natural selection to select. Some examples of hybridization and epigenetics working together at the same time. The quick brown, using English grammar as an analogy, the quick brown fox jumps over the lazy dog, period. The slow golden fox jumps over the crazy dog. If we then combine that with the slow red fox leaps over the lazy dog and the fast red fox jumps over the big black dog, through hybridization, we have double the amount of information, double the amount of computer code that we originally had. So there's more opportunities for the other mechanisms to select and turn on and off and move around, transpose different genes. Through epigenetics and editing, we eventually can get down to The slow brown fox and the red fox jump over the lazy golden dog, the crazy dog, and the black dog. Again, this is following the rules of English grammar. You couldn't have predicted how this would have turned out at the end, but it's not random because it's following the rules of English grammar, just as the mutations that give rise within evolution follow the rules of genetics, chemistry, and biology, and physics, ultimately. So, to summarize, Thesh, I had to if we list these in order of gradual to sudden and common to rare, epigenetics is common, followed by transposition, horizontal gene transfer, hybridization, and symbiogenesis. This also gives, so as we move further down, these things become more sudden, more instantaneous changes. You can get drastic changes in an organism in one or just a few generations of the species, but it's very rare for that to happen. So hybridization, hagfish becoming, or hagfish coming from whatever those other
Tunicates.
Thank you, tunicates. That's rare, but it's very sudden. This is where the idea of punctuated equilibrium comes from, where things don't evolve until they have to. They're basically the same for very long periods of time, and then very quickly lots of things change, and you get new species out of that. So this gets back to my original idea, is that evolution is easy. Like, quote-unquote easy. I mean, there are these different mutation mechanisms to allow the code to get moved around in beneficial ways, to recombine in beneficial ways, to get transferred between species in beneficial ways, and then all you got to do is kill off things that don't work and then allow things to live that do work. Another thing I've become aware of in this is that evolution can be driven very quickly. You can have a petri dish of cells, and you give them enough nutrients, give them everything they need, they're happy, and mutations are incredibly rare. They're not mutating, they're not changing. However, if you introduce a mild toxin or you starve them by not giving them enough food, all of a sudden the mutations go through the roof. Things are mutating like crazy, trying to figure out combinations of code that will allow them to survive. Most of them will die, but some of them will get the combinations of code correctly through the mechanisms we just talked about. These non-random mechanisms, they're just trying out different combinations of code to find something that will work. Eventually they do, and you evolve some adaptation for either the toxin or the environment. This is incredible because this means that the cells, through mechanisms that are not understood, and if you could answer these, you get a PhD and a Nobel Prize in biology. How do the cells know how to reprogram themselves? How do they know, how do two species of cells know how to transfer information through a pelus in, for example, horizontal gene transfer? How do they know to transpose their DNA? I don't think those answers are currently known. Those are open questions in biology.
Is the mere principle of survival unknown? I mean, how do they know that they're being threatened, and if they don't do something, they'll get destroyed?
Yeah, I don't think that's known. So these are open questions in biology. We know what happens, we know what causes the mutation, but we don't know how the cells, and really within the cell, like the transcriptase, which is a protein enzyme or something that goes along, it rides along the DNA like a roller coaster, and it copies the information, or it can fix the information that's there. How does it know to start reprogramming the cell? It's like these molecules are smart somehow. That's really cool, but it's also a mystery and an open question in science. Andrew.
So you're talking about a little bit further back about the genes that would like switch to different things when introduced to different environments. You're talking about the famine.
Yeah.
Place.
Epigenetics. That's epigenetics.
Epigenetics. So did the actual genes of the people in that situation, did they switch in the people that were just introduced to that situation before they even reproduced or anything?
Yeah, so the people who were being starved in the famine, some of them developed epigenetic changes.
So their genes just switched just by being introduced to that
Right.
situation.
Now, their code didn't change. Some code just got turned on or off.
Oh, okay. So some that would have been dormant turned on.
The same thing happens if you get calluses on your fingers from playing guitar. That's an epigenetic change. Your body is turning on and off certain genes. The information is not changing. You're not mutating per se, but you're just turning on and off different parts of your code. And so do you have calluses from playing guitar?
I do.
Yeah, okay. So that's an evolutionary epigenetic change to protect you from those strengths.
What causes the little bridge, the pelus, in the first place?
I'm not sure. The two cells somehow in their intelligence know to do that. I don't know. But that's a great thing to Wikipedia when you're home. You know, these things are observed to happen. We know they happen, and somehow the cells are smart enough to know how to do that. Yeah, Greg.
I have slow, gradual evolution of organisms, I find totally believable. Things that I have a hard time swallowing are things like the wing, where sure it could have slowly evolved, but there's no useful benefit of the wing until it reaches a certain level of
Right.
complexity,
Like the bridge halfway
utility.
idea.
That type of thing I've never found adequately explained.
I mean, just through hybridization, we showed how you can go from two species without a jaw, for example, to all of a sudden having a jaw. So you could imagine something similar happening for the development of a wing. A wing could develop very quickly within a generation or two.
Right, but
And the wing, even a proto-wing, a bridge halfway, so to speak, could still fulfill another function before it fulfills its final function. And I know that...
I mean, with something like a jaw, though, you can still eat. You know, that's not preventing you from eating. It may not work as well as it could eventually, but I think something like a wing, it serves really no purpose. That's where I find it. I mean, a wing has to be very specifically designed to serve any real purpose.
We still have a lot of birds that have
There's a lot of
wings,
flightless
but don't
birds.
use them, yeah.
Right.
That's a wing halfway, not serving a purpose. It's probably a mating thing. Like, look at me, I'm big and attractive.
Right. I can see an animal devolving such that it can survive on other things without the wing, but to actually develop that originally is where something like
I mean, there's definitely more research to be done. I mean, you can imagine like a T-Rex with its tiny little, like, these are probably vestigial from when the earlier dinosaurs were literally on all four.
Right.
I mean, these serve no purpose for a T-Rex. Yet, these then later evolved into wings that didn't serve flight, but could maybe help you glide from tree to tree. Things that eventually could help you fly. So I'd be careful saying, well, you can't go from no wing to wing. We just showed how a vestigial arms on a T-Rex went from something apparently useless to becoming flight wings for early dinosaurs and then birds. I don't know the more satisfying answer to your question, but I suspect those mechanisms exist.
So take me back to Genesis and explain to me how Genesis connects to the vestigial scale on us.
I don't think it does, because Moses had no concept of that whatsoever.
I'm trying to obviously grapple with the whole idea that
Sure.
God created us,
Yes.
but this is how he did it.
Right. So I do believe that we are evolved from the same ancestors as other primates. Nevertheless, God created us with purpose, teleology, that no science experiment can point toward. I'm going to speculate on this really fast in the last couple minutes. So I would love to dive into more evolution and biology, but hopefully I can do another Sunday school class where you can look this up. So Adam and Eve, a lot of ideas. I'll just tell you what I believe. I believe they could have been historical people out of an original human population. God having called both of them out of a larger human population to live in unique relationship with Him, somehow mysteriously giving them a spirit, a way to outlive their death, perhaps even giving them the supernatural ability to not die initially. I might go there speculatively, and having the historical Adam and Eve represent God to the rest of creation, whereby there was later they were created to live in a relationship with God. They chose at some point to rebel, whether that was literally eating an apple out of a tree that they weren't supposed to, I don't know. But there was a conscious act of rebellion against something God had said to do that introduced sin into the world and necessitating our redemption through Jesus. If you understand the imago Dei, the image of God implanted in us, I think theologians have talked about that being several things. That's us having a soul. No other animal that we know of has a soul, a spirit to dwell with our animal nature, are being created to live in relationship with God, us representing God to the rest of creation. I think that's a big thing. Our environmental stewardship and how Adam was called to tend the garden, that's pointing towards our role in understanding creation and taking care of the natural world. Having Adam and Eve be the first humans only in a spiritual sense and not in a genetic or physiological sense also solves some problems, because Genesis also talks about, after Cain killing Abel, saying, people are going to kill me. Where do these people come from? The Genesis text doesn't address that. It also, talks, also to frankly explain the amount of genetic diversity in the current human population. You can't have two people. You have to have an initial human population of at least 10,000 to explain the amount of genetic diversity we have in humans. Greg?
So with Adam and Eve, the forming Eve from the rib of Adam, could that have been some kind of hybridization type thing?
I suspect not.
I was actually just kind of thinking about that. Because you're talking about
Fairly certain women originate from a different species.
I'm not going there.
you're talking about how like some you have cells in a dish, and then you introduce like some sort of like starvation or some sort of event that makes it really what are the words I'm trying to say? You know what I'm saying? Yeah, some sort of event that kind of forces them to do certain things.
Mm-hmm.
Well, Genesis describes that because it talks about how he made Adam, and then he's going around, he's naming all the animals, and then it says but he found no suitable mate for himself. So in a way, that's a sort of event that would force genetic mutation in a way. Think about it.
Interesting. I have to think about that more.
Yeah.
Where I see this as particularly beautiful is this God calling out two individuals from an original human population foreshadows God calling Abraham out of an initial human population, ultimately God calling Israel, and then God calling us, the church, to live in relationship with him. It's just a continuation backwards through the scripture of this idea of God choosing to live in covenant relationship with chosen people. And these original two spiritual humans could represent God in some beneficial way. I mean, it's through Israel that God is blessing the world. It's through now Christ and the church that God blesses the world. It was through these two original individuals that God was going to bless the other 10,000 humans living at the time. Now, did these other 10,000 people acquire a spirit at the same time and become, I don't know. Is this passed on genetically? I don't know. There's a lot of mystery here. This also foreshadows Jesus talking to Nicodemus, saying, you must be born again. In some ways, this goes against the Lady Gaga song, I was born this way. Yeah, you were born that way. Yeah, your evolutionary heritage, you evolved this way, but you need to be filled with the Holy Spirit, transcend that, be born again, to live in a way that's pleasing to God.
All right, well, given the spiritual aspect, if you could, you may have already done this, I didn't follow you. Points 1 and 2, if you could dive into that and explain
Death
that.
before the fall, Romans 1, just talking about human death, not animal death.
No, I mean,
Okay.
there's a huge spiritual distinction, I think, between Adam and Eve and post-fall mankind. So what is the evolutionary explanation to the introduction of sin, let's say? I don't know that Adam and Eve had the capacity for sin, but hadn't exercised it, and they exercised it. I mean, that's a big gap, it seems to me, in man's destiny.
I don't see that manifesting itself biologically, if that's what you're asking.
Right, so, but you're talking now in God's role, in the spirit, we have a spirit, we have a soul, you know, we're different than the other 10,000, so we experience sin in a profound way that leads to the subsequent steps of chosenness and reconciliation and ultimately salvation. So, you have to have some kind of split at the very beginning between Adam and Eve as intended and Adam and Eve as they were.
Yeah.
And I don't know, I'm just wondering if you personally or professionally have any thoughts on those lines.
Not off the top of my head.
Well,
So sin was inevitable. It's just a natural.
I'm not saying that. No.
Okay,
no.
well, so what are you saying about sin?
well, before like God.
I would go with what Scripture said. There was a conscious choice at some point to rebel against God.
Okay, so you're just, okay, all right.
That's all I'm saying.
Okay.
Before God came along,
Thanks, Greg.
came to the picture, we wouldn't have even recognized that sin was sin. So until he came along, there was no distinction.
Right. Romans talks about that. Where there is no law, there is no concept of sin.
Yeah.
Right. I want to be sensitive to the time. It's time to, if you're going to the 11 o'clock service, we're getting close to that. But a few big bullet points out of this. There's a lot of animal suffering that goes on in evolution. Things have to die for natural selection to work. And I don't know. I would have to say that animal suffering is not good or bad. It's amoral. I don't know. Ultimately, I don't know. And like Job, we may never know this side of our own future resurrection before this side of Christ's second coming. A few other examples, a few other points I want to take away, that God rarely acts swiftly, but he seems to savor the process of creating over long periods of time and doing things in relationship with his creation, where his creation, where it's able, brings about his own creative goals. And this way, creation is a servant of God, but also a colleague of God, to co-create. And this anticipates us being made in the image of God, where everything that God is, we do on a micro scale, except that we're also fallen. And this whole idea is that God designed a process that favors cooperation and fosters development, I think is morally compelling. So, but there's but there's tons of speculation we can do as to how to reconcile these ideas of our evolutionary descent and our spiritual status. And it's ripe for theologians and people smarter than me to speculate about it.
Thanks.
Yeah.
Thank you, Brent.
You.
Yeah.
I like it.
Yeah, plenty smart enough for us.
I'll say.
Thanks, Hillary. If you ever go to Bozeman, go to the Museum of the Rockies. There's tons of fossils there. That's a Triceratops. And this is a Deinonychus eating a Tenontosaurus. There's a real dead Deinonychus.
It was just a piece, I